Dear all, welcome to our channel. Today we are going to start a new lecture of series that is about the biochemistry, and in this series we are starting with the amino acid, peptides and the protein. In this lecture you will learn about the amino acid structure, the what are the different types of the amino acids, how they are classified, how the amino acids are combined to form a peptide and then to a protein. So in this lecture we will discuss about the general structure of the amino acids, how the amino acids are classified into different groups based on their based on their R group. So that all these things we will discuss into the this lecture. Let's start with the protein. So because the amino acids they make the proteins, so we can say proteins are the polymer of amino acids. So proteins are the most abundant, it is the most abundant biological macromolecules, and it occur in all cells. Like in a body you will find the presence of the protein in all type of the cells and all part of the cells, because it is the major functional biomolecules which perform various type of the function into the cells. So it is found in all type of the cells and all part of the cells, means all the organelles will contain this protein. So if you will say what the protein perform various type of the functions in a organism, like it catalyze the metabolic reactions. There are various metabolic reactions occur into the body. So all these metabolic reactions require the enzymes, and mostly the enzymes are made up of protein. So all these catalyzing the metabolic reactions are conducted with the help of the protein. And other DNA replications, like some molecular biological reactions such as DNA replication, DNA transcription, translation, they also involve various type of the transcription factors and translation factors. They are also made up of protein. So they also help in these molecular biological reactions and respond, like third, that is responding to the stimulus. Like hormones are bind to their receptor, then they respond, their stimulus is passed to the cells. So in this way the receptors are also made up of protein. So in this way they are helping into the response to the stimulus, providing structure to the cells. Like many structural proteins such as keratin, actins, which provide the structure to the cells and the organism. uh, the structure to the cell, that like keratin, myosin, these all are also a protein. And in the organism the various structural proteins are present, that is uh, like in the hair, horn, they are also made up of proteins. And transporting molecules, so that molecule are transported from one place to the another. Like example you can take is the hemoglobin, where the hemoglobin transport the oxygen from one part of the body to the another part of the body. So all this conducted with the help of the protein. So proteins are having a very diverse function in an organism, and it perform various type of the functions. So that's why these are the most abundant biological macromolecule. Now proteins are occurring great variety, like different, as we have seen that they are having involved in various type of the cellular function. So they are found in different types, thousands of different types of the proteins you will find in a cell or in an organism. So and these thousand different type of the proteins, they also vary in the size. Like they can be of a small peptide of like 3 to 4 amino acids or 10 amino acids, to the huge polymers where thousands of amino acids are linked to form a protein. So their size, protein size also vary from few amino acids to the thousands of amino acids. And how these proteins are differ, these proteins, like they are performing a different function, so they also differ into their sequence. So we have to remember that proteins are made up of 20 major common type of the amino acids, and these proteins are formed by the amino acids. Amino acids are combined in a sequence form, and this sequence is determining their structure. And how this structure sequence is coming for the protein, like which amino acid will combine to next, then this is all dictated by the nucleotide sequence of their gene. Like the DNA that is having a sequence, and then DNA is having a sequence over a particular gene, and that gene is then transcribed from RNA, mRNA, and then from mRNA a protein is synthesized. So this protein is the sequence of the amino acid that form a particular protein that is determined by the gene. So then after this, these proteins are synthesized, they are folded according to their sequence, and then they form a specific three-dimensional structure, and their three-dimensional structure that determine their activity. So all this, the protein functions, and their that is determined by the sequence of the amino acids, how they are arranged, and this sequence of the amino acid is determined by the nucleotide sequence that is present into the DNA. So next, that all proteins, like if we are saying that 20 amino acids, the proteins are made up of 20 set of the amino acids. So whether they are the same type of the amino acids are found in bacteria, plants, fungus, or human, monkey, all type of the other organism. So you will find that all the proteins, whether they are from the bacteria or the simplest form of the life, that is bacteria, or the viruses, or the most complex form of the life like mammals such as human, monkey, whales. In these all these animals, all these organisms, the proteins are made up of same type of the amino acids. Like all these 20 amino acids, they makes the different type of the proteins, and these amino acids are then linked in a characteristic linear sequence. As we have discussed, that linear sequence, that sequence is coming from the DNA. So the cells can make a proteins with different properties and activities by joining the same 20 set of amino acid in different combinations and sequences. So in this way, we can say these proteins are made up of same set of 20 amino acids. So proteins are the polymer of amino acids that we have discussed, that proteins are made up of amino acids. There are 20 amino acids which makes all types of proteins and with diverse functions, and the proteins, these amino acids are same in, like from bacteria to the human being, like from a simplest form of a life to the complex form of the life. This same amino acid make the different the same amino acid make the protein in all these life forms. And but how these proteins are differing? They are differing according to their sequence, how they are arranged in a sequence. And like we are saying that amino acids are joined, so these amino acids, they are like joined. Like if this is a one amino acid 1, this is amino acid 2, this is amino acid 3, and so on. So these amino acids, they are linked by a bond. That is, the bond is named as peptide bond, and we also sometimes call it as amino acid residue. Once a amino acid is combined in a protein, it loses its water, and then it is called as a term as a amino acid residue. So if we will hydrolyze this protein, then hydrolysis of the protein will break down this peptide bond, and we can get the constituent amino acids. How these proteins are made up of, like what are the different amino acids which are involved into their formation. So that we are saying that 20 different amino acids are commonly found in the proteins. So the first amino acid that was identified was asparagine, that was identified in the year 1806, and the last was threonine. Threonine was identified into the 1938. So it takes lots of time to identify all the 20 amino acids. So let's see with this picture. In this picture, you can see that these amino acids, we can see this picture that these proteins, the different function of the proteins. So you can see that this first picture is about the firefly. Second is for the erythrocyte, and third we are seeing the structure of a horn. So this first picture, you can see the light which is produced by the firefly. It is this firefly produce a light. So this light is produced due to the reaction between a protein, that is luciferin, and a ATP. So this protein, luciferin and ATP react and form. This reaction is catalyzed by an enzyme, that is the luciferase. So this protein, so this light production is due to involvement of a protein. Similarly, in the picture B, that is about the erythrocyte. Erythrocytes are found into the blood, and these erythrocytes contain a pigment that is named as a hemoglobin, and this hemoglobin is containing a protein, and this hemoglobin is helping into the transport of the proteins from one part of the body to the another part of the body. So this is helping into the transportation of the oxygen. Another protein, that is the, you can see this is the image of a horn of a rhinoceros. This protein is made up of keratin, and the protein keratin is found in all the vertebrates. It is a chief major component of the different structural proteins, such as the hair, scales, which are found into our body. Hair, you find the scales which are found into the fishes, and horn, which is here you can see this. All are made up of a protein, and this protein which is involved here, that is the keratin. So we can say that by seeing this picture, and these proteins are having a wide diverse function in a body. Like it is involving in a light production, it is involving in a metabolic reactions, it is involving in a transportation of the biomolecules, it is involving in a structural protein. So they are having a very diverse function, and amazingly, these all proteins are made up of same 20 amino acids. Like in a firefly, if you will see, it will have the same glycine. In the here also the same glycine is here if it is involved, and here the same amino acids will also find here. So if you will isolate the amino acids from these three different source, the similar type of the amino acid we will find. So now we will see how these amino acids are named. So these amino acids are named based on the source of the identifications. Like asparagine, that was identified first in the asparagus, thus they are named as asparagine. Glutamate, it was identified first from the wheat gluten, so it is named glutamate. Gluten, glutamate. Tyrosine was first identified from the cheese, and the cheese in the Greek is called as tyros, so it is named as tyrosine. Whereas the glycine, it is having a sweet in nature, so here it is named according to their nature, because glycine is sweet in nature, and in the Greek it is called as glycos. So thus based on this it is called glycos, so they are called as glycine. So now we will see that how now we will discuss about the structure of the amino acid. These 20 amino acids, what are their structure? As we are saying this, these proteins are made up of same set of 20 amino acids, then they combine into different sequence and makes the different proteins. So what are these amino acids, the different type of the amino acids making the different type of the proteins. So but you will see that amino acids share the common structural features. All the amino acids are having a similar type of the structure, that is the backbone structure. You can see here, that is a carbon, central carbon. To this central carbon a carboxyl group is attached, and amino group is here also attached. That's why they are called as amino and carboxyl group. Both group are present. That's why they are called as amino acid. And you can find the carboxyl and the amino group are attached to the same carbon. That's why they are called as alpha amino acids. And here H group. So this is the common structure that you will find in all amino acids. But how they are differing? They are differing according to their R group. This R group will differ into all type of the amino acids. So this is the general structure of the amino acids. So what we are saying, this structure, that amino group, carboxyl group, the H group, and this carbon will find in all amino acids, and the different amino acids will differ according to their R group. But here is an exception, that is the proline, because in case of proline it is a cyclic amino acid. Here the this R group is covalently bind to its amino group. Due to this, it is not having this type of the structure. So here it is called as alpha carbon. So this amino group and the carboxyl group is attached to the same carbon. That's why it is called as alpha carbon. Here it is a structure of the another amino acid, that is lysine. In the case of lysine, you can say there are additional hydrocarbon chain is found, and here it is another amino group is attached to the epsilon carbon. So how we are saying this is epsilon carbon, how their additional carbons are named, that is based on the, so it is the alpha carbon, then the next to this alpha is we are saying the beta carbon, and then next to the beta we are saying the gamma carbon, and delta, and then epsilon, and so on. So in this way these additional carbons are named in amino acids. So this is the structure of the lysine. So what we are saying, these amino acids are having a common structure, but their R group is different, and that R group is different into their structure as well as in their electric charge, and their measure is the property to solubilize in the water. Based on this, the amino acids, all the 10 amino acids has been characterized into polar or non-polar, whether they are polar or non-polar. So this R chain will determine whether they are able to solubilize in the water or non-solubilize into the water, and their structure, their chain size is also differ, and their charge is also differ, and all these properties are making the different type of the amino acids, and these amino acids presence is making a specific type of the protein. So we have seen that there is a chiral carbon. So we are seeing in the last slide, we have seen that there is a carbon to which all the different groups are present. So such type of the carbon where all the four bonds are, all the four valency are occupied by the different groups, such type of the carbon is called as chiral carbon. So here we can say that this chiral carbon is present into the amino acids, but because there is an exception, there it is a glycine for where the R group is H. So here if in the case of glycine R group is H, then all the four to the carbon, all the four groups that are not different, here these two groups are similar. So due to this, glycine is an exception which is not containing the chiral carbon. Fourth carbon is H, so it is not the having a chiral carbon. So the where it is a like in stereochemistry we learned that where to a carbon when there is a four different groups are present, it can occupy a spatial arrangement in the space, and thus amino acids have a two possible stereoisomers, that is the D and L form. So these amino acids, these groups can arrange in a two different form, that we will discuss in the coming slide, two different form. So due to this, these amino acids can be called as D amino acid and L amino acids. So that is the stereoisomerism in the amino acids. That is, we are showing the analogy with the glyceraldehyde. Like in the glyceraldehyde, this is the L glyceraldehyde and this is the D glyceraldehyde, that we call as D and L system. How we are saying this is the L glyceraldehyde and the D glyceraldehyde? This carbons align vertically with the chiral carbon into the center. If we are seeing to the chiral carbon, this hydroxyl group, it is on the right side in this glyceraldehyde. So then we will say it is a D glyceraldehyde. What we are saying, if the hydroxyl group is on the right side, then we will say it is a D type of the form, stereoisomer. And here if the hydroxyl group is on the left side, then we will say it is a L glyceraldehyde. So that is about D form and the L form. So if we see the analogy with the amino acids, similarly here the amino group that is on the left side, so then it is a L amino acid, and here amino group is on the right side, so this is well called as a D amino acid. So in general, in a protein, generally the L type of the amino acids are found. So how we can identify if the amino group is on the left side, then we will say it is a L-alanine, and if it is the amino group is on the right side to the chiral carbon, then we will see is a D amino acids. D amino acid. So like L-alanine and D-alanine. So there the amino acid is not differing into their chemical nature, chemical like in an atoms, but the spatial arrangements only differ here. So that makes the different type of the amino acids, and the proteins which are found into the like in our biological system, they are also very stereo specific. So amino acids in the proteins are L stereomers. So you will find that all the proteins where the, you know, they are the amino acids that will always in the L form, exclusively in the L form. If you will see the proteins, they are always made up of the L form. The D type of the amino acids are not found in the proteins. They are always only found in some like cell wall proteins and the plant cell wall protein and certain antibiotic. In general protein, you will not find the D amino acids. And if there is a D amino acid is found, then in place of the L amino acid, the enzyme will not catalyze the reaction. If you will replace a D amino acid with a L amino acid with the D amino acid in a protein, then and allow the enzyme to react, then it will not react. That's these enzymes are also very stereo specific, because all these in our protein, these exception of some bacterial cell wall protein and then some antibiotic proteins, these proteins are L stereoisomer. So what we are saying, cell is specifically synthesize the L isomer of the amino acid. Cell कौन सी टाइप की अमीनो एसिड को सिंथेसाइज करती है? दैट इज द एल टाइप ऑफ द आइसोमर, because the active site of the enzymes are asymmetric. Enzymes catalyze the reaction that to be stereospecific. Thus, they will catalyze the reaction to the protein which are having the L isomers. If the protein is having the D isomer, like if it is a protein is having a D-alanine, so it will not. And it has to be catalyze a reaction based on this amino acid. It will not catalyze that reaction. So that now we have learned that what is the general structure of the protein, and the L amino acids are exclusively found into the proteins, and cells synthesize the proteins which are made up of L stereoisomers. So now we will see, and we are saying that the proteins are differing according to their R group. R group will determine their chemical nature, and in particular R group is very into their polarity, like how this R group interact to the water, whether it is able to solubilize into the water, how it is forming the hydrogen bond with the water at near biological pH. Based on these characteristics, protein has been broadly classified into two groups, that is polar and non-polar. And later on, based on the aromatic, whether it is the aliphatic side chain or the aromatic side chain, they also has polar or non-polar. So based on this, they has been classified first polar or non-polar. Later on, if they are having a non-polar, whether they are having an aliphatic side chain or aromatic side chain, they have classified based on this. Next, polar, then whether they are uncharged or charged. So based on this charge, then if it is a positive charged or negative charge. Based on this, there are 5 classes of the amino acids, and if you will broadly say, this is based on the characteristics of the amino acid to interact with the water, whether they are polar in nature or non-polar in nature. So that we have discussed the steric relationship of alanine with the glyceraldehyde. So now comes to the classification. So as I've already told that classification of the amino acid is based on the R group. The skeleton carbon will same in the all amino acids, and these amino acids will differ only into their R group. That they are having a non-polar aliphatic R group or polar R group. So non-polar, they are having a two type, aliphatic R group and the aromatic R group, and polar, they are charged or uncharged. So if it's charged, then whether they are containing a positive charge group or a negative charge group. Based on this, these amino acid has been classified. So this in the next lecture we will discuss about this, the the classification of the amino acid based on their R group. Thank you for watching this lecture. We will meet into the next lecture where we will discuss about the classifications of the amino acids based on this R group, how they are differing into their structure. So that we will discuss into the next lecture. Thank you for watching.